Hello everyone. Today I'd like to talk about potential brain tumor therapy by using an intracellular combinative infrared laser exposure. And this work was collaborated with many people. Professor Chen is from E department, and Professor Dr. Paul is a doctor from Department of Radiation Oncology, and Chen is a surgery doctor from the Department of Neurosurgery product department, and Professor Gundersen is my advisor. And today's talk, because glioblastomas is a aggressive cancer of the brain, and we know that the cure rate is pretty low. And in this work, we try to investigate hyperthermic ablation of glioblastoma multiforme tumor. We use by irradiating intracellular carbon nanotube with near-infrared light as a potential therapy. And later, I would like to show you how serious glioblastoma multiforme is, then tell you why we choose the near-infrared radiation, and show you how do we know that carbon nanotube will go into the cell. Then the experimental result can show you the response of the cell with and without carbon nanotube and under the near-infrared laser exposure. Then I will do a conclusion in some future work. Okay, so this is the brain and this is the tumor. And we know that the glioblastoma is highly malignant, and rapid infiltrating means it will migrate to the normal brain cell quickly. And we know that it affects approximately 17,500 patients annually, and only 10% of patients can survive more than 1 year. And we know that if we cut the tumors, we cut this, but it will reoccur in the resection area after sometimes because that maybe during the time of surgery, then the brain tumor already invaded to the normal brain cells. So it's the reason that the survival rate is very low. Okay, in this slide, from here, we can see that the tissue, this is the human tissue but not the brain. We can see here is an open window around 700 to 1,100 nanometers. It means that the human tissue is transparent at this range. But we try to measure the absorbance of the carbon nanotube. You can see this is carbon nanotube in PBS solution. PBS is phosphate buffer solution. And we can see here is a peak at LOL, but if the cells in PBS solution is very flat. So it means that if we use this LOL nanometer laser to expose the cell and the carbon nanotube, only the carbon nanotube will produce heat, but the cell will not be cooked. Okay, and this slide is our setup. You can see we use the LOL nano dye laser. Here is the dye laser. The light comes here and the mirror. And this is the cover glass chamber that we culture the cell inside. And we measure the power. The power of laser here is around within 1.2 centimeters square. The power is 1.5 watt per centimeter square. And because we want to see the cell without carbon nanotube under near infrared laser, so we try to change the exposure time to find out the optimal condition. So we use several different exposure time. And we also use an indicator called propadian iodide, and it is only taken out by the cell if the cell membrane is not complete. So it means that the cell is dead. So if the cell is dead, the PI will go into the cell and show us the red fluorescence. Okay, so first step, carbon nanotube, because carbon nanotube we use is 1 nanometer in diameter and 200 nanometer in depth. But because carbon nanotube is no visible fluorescence, so we need to attach a dimolecular on its surface, then we can see the fluorescence under microscope. So the way we use it is that first we create a COOH group on the surface of carbon nanotube, then EDC can bind the fluorescein and the carbon nanotube together, so finally we can get a carbon nanotube fluorescein conjugate. Then we put this kind of conjugate into the cell, then observe under microscope to see if the inside the cell really shows the fluorescence but not outside the cell. This is the microscope that we use, is the EP fluorescence microscope. And how do we know that the carbon nanotube fluorescence really inside the cell but not outside the cell? So we use a system called Z-Stack 3D image systems. And this system is that it can take the image at different kind of focal plane, then finally put them together to form a 3D image. So these stack images are a series of image of a specimen taken in a sequentially focal plane. And we also can change the distance of focal plane to get the higher resolution 3D image. So okay. So the right one is the U251 glioma brain cancer cell. Because the GBM cell was derived from the astrocyte, this kind of normal brain cell, so it will be interesting to see if only the brain cell will uptake the carbon nanotube, or also the normal brain cell will uptake the carbon nanotube. And so I try to get the 3D image of the intracellular carbon nanotube from two different kind of cell. And just like this is the top image, just the just from the top plane. So finally, we can see that this 3D image really shows us that the carbon nanotube fluorescent conjugate go into the brain cancer cell and the normal brain cell. Okay, so from this slide, and we can see the right one is just the brain cell without carbon nanotube, and the left one is the brain tumor cell with intracellular carbon nanotube fluorescence. And before this, I watched the carbon nanotube fluorescence in the median, so it's pretty sure that only the carbon nanotube the carbon nanotube fluorescent conjugate really inside the cell but not outside the cell. And we take this cell under 8 meter laser exposure, and after 9 minutes exposure, it really tells that if the brain tumor cell with the carbon nanotube fluorescent conjugate, it was killed. The red color is from the propylium iodide. It's an indicator to tell us if the cell is alive or not. So we kill the cell, the brain tumor cell. But maybe you will question that, how do you know that fluorescence will not have any other effect? So in this way, we repeat the experiment, but we will replacing the carbon nanotube fluorescent conjugate by just single wall carbon nanotube. And you can see this is the next slide. This is the same brain tumor cell, but we just replace the carbon nanotube fluorescent conjugate by single wall carbon nanotube. And after 9 minutes exposure, the brain tumor cell with intracellular carbon nanotube was killed. So it will be more interesting to see if there are some selectivity if the normal brain cell with intracellular carbon nanotube were killed or not. But so we replacing the brain tumor cell by the normal brain cell. But unfortunately, there is no difference if the normal brain cell with the intracellular single well carbon nanotube, it will be killed. Okay, so finally, my conclusion is that carbon nanotube can transport covalently attached molecule across the cell membranes, and taking the advantage of the fact that carbon nanotube can absorb and on nanometer radiation much more stronger than brain tissue. We demonstrate that this may be a way that we can kill the brain cell and brain tumor. And in the future, ongoing work is to try to see if we can targeting the brain tumor with carbon nanotube, but not the normal brain cell. And in this way, we can just only kill the brain tumor, but not the normal brain cell. Okay, so this is my talk. Thank you.